Okay, class. So now we're going to do part two, cancer. And just as a reminder, this won't be on the exam, but you guys, I think, will find this very useful regardless. And one of the caveats that I have to throw out here, I'm not a cancer biologist, but your book does a pretty good job of highlighting some of the big stuff. So I will sort of stick to the cell biology parts of it because really cell biology has benefited uh, sort of greatly from studies on cancer cells, right? The just trying to figure out what goes wrong in a cell when the cell becomes a cancer cell has been, just an intense area of research for, 50 years or so, like or more. And ultimately, what happens in cancers are a variety of regulatory pathways sort of are broken. And, the idea is if we understood cancer and all the regulatory pathways, then we can find things to pinpoint and treat and develop great chemotherapies. So, really understanding cell biology is a key to a lot of this. And one of the reasons that cancer is so prevalent is that 10 to the 14th cells acquire a mutation every day. So we're just getting bombarded all the time with things that damage our DNA, and even just just copying our DNA, it makes on average 3 mistakes every time. So, you can sort of see how cancer, and we'll see this later, it's really not a matter of if, but when cancer will arise. So we're all susceptible to it. Um, so here's a nice little CAT scan of metastasis. Okay, so cancers are immortalized cells, and they can divide indefinitely. A benign tumor or a neoplasm is localized, but a malignant tumor will spread throughout the body. And in this case, you're looking at a patient with non-Hodgkin's lymphoma, and you can see that the cancer has spread to all kinds of different tissues and organs. Um, Now, historically, cancer has been sort of defined by the tissue in which it originates. So, breast cancer, prostate cancer, colon cancer, lung cancer, you name it, leukemias, blood system. But one of the things is that cancers, they're now finding through sort of personalized medicine and precision medicine that the genotypes of the underlying cancer is what really matters, right? So you could have one patient, female with breast cancer, and the underlying genotype of that individual's cancer may be more similar to a male who has prostate cancer if you looked at all of the genetic lesions and the proteins that have gone awry. And so that makes a big deal in the treatment options, because you don't necessarily want to go into a clinical trial and say, I have this drug to treat breast cancer, and and it's only for breast cancer, when it could be very effective as a treatment for a different form of cancer with the same genetic lesions. So there's a big, huge push right now to understand cancer on the genome level, to find out what the mutations are. There's a big NIH initiative called the Cancer Genome Atlas, where they're basically doing that. They're looking at the expression profiles and the underlying DNA sequences, the proteins that are mutated, and all of those things, and they're trying to get a better classification of cancers other than in the inherent tissue that it arises in. Okay. I hope that made sense. So just some terminology that you need to know: benign versus malignant. So here's just a normal duct. Benign tumor is one that hasn't spread and hasn't gone anywhere. However, when it starts to develop all of the necessary things to escape, it becomes a malignant tumor, and then it can spread. Now, tumor growth. One of the problems with treating cancer oftentimes is that when you detect it, it's already very late in the game, okay? Because so many cells have to first arise in order for it to be detectable. So here's just basically a graph showing the tumor cell population as it's doubling. And when a tumor is first visible on an X-ray, there's already a substantial number of cells. Okay, and the first time you're going to notice it as an individual, whether or not you can feel it, will be 10 to the 9th, and you don't really have that much long to go before the patient will eventually die. And so early detection is really the key. And there's a big interest in finding biomarkers and things like that are circulating so that you can get early detection. Because the earlier you detect it, the more effective most of the treatments are. And I'm sure you guys have all heard that at some point in time. Okay, so here's the Philadelphia chromosome. We covered this before when we were talking about chromosomes and translocations. But the Philadelphia chromosome is actually a translocation between chromosome 9 and chromosome 22. And so what you end up getting is this tiny little Philadelphia chromosome and then a translocation over here. And this generates a new fusion protein called Bcr-Abl, and it occurs in CML, or chronic myelogenous leukemia. And there's a great treatment for this. This used to be one of the most deadly leukemias, but now there's an incredibly effective treatment called Gleevec, which we'll look at the sort of the end of the lecture. But this is just one way that a cancer can arise. You'll get this translocation, and all of a sudden you'll generate a novel protein called Bcr-Abl. Now, cancer originates from a single cell. And so you can sort of follow that, because if you have like an egg and you have your maternal X and your paternal X, this cell ends up undergoing random X chromosome inactivation. So it's not like imprinting where you'll get a paternal copy silenced or the maternal. In the case with X inactivation, it's random. Either X can be inactive, but every cell is going to have a different is either going to inactivate one or inactivate the other, and that occurs from the X inactivation center. So ultimately, when the cell starts to become an embryo, you'll have different inactive X's. Either the maternal X or the paternal X will be active or inactive. And then you'll have your adult tissue, but in the cancer and in the tumor, it's always going to be the same X that's active, either the maternal X or the paternal X. So they can basically use that to sort of determine that, well, it likely came from the same cell. Otherwise, you'd have a mixed population if it was coming from many cells where multiple different X's and from the paternal or the maternal would be inactive. I hope that made sense. Okay, one other thing with cancer, and I sort of alluded to this before, it's really an age-related disease. Although you can have childhood cancers, they're oftentimes inheriting a tumor gene from their parents, or something, really crazy goes awry. But in general, you have 10 to the 16th cell divisions in the lifetime and 10 to the -6 mutations per gene. So what that basically means, if you do the math, is that every gene has the probability of getting 10 to the 10th separate mutations, okay? And so what ends up happening is, as you increase in age, the incidence of cancer also increases. So it's not about if, it's about when for most people. Some people can live an entire lifetime and it doesn't happen, but it's a game of chance, right? And one of the reasons too is that it's typically never just a single mutation, right? There's usually multiple things that have to occur. So you kind of have to, it's almost like playing Russian roulette. Where eventually it's going to happen. And if you live long enough and you don't die of something else first, you're probably going to get cancer. Now, cancer also has to do with how you're exposed to various carcinogens. So in this instance, here's just a graph of a carcinogen to naphthylamine, and depending on how long an individual is exposed to a certain carcinogen will sort of dictate how often they're going to get cancer. So in the case with 5 or more years of exposure to this, after a certain number of years, close to 100% of all of the people, would develop cancer. Whereas if you have a shorter exposure, 3 to 4 years, some people can escape it. In less than 2 years exposure, only a few people get it. Okay, so this is the percentage of people with bladder cancer. So once again, the more you're exposed to specific carcinogens, the more likely it is that you're going to develop cancer. Here's just an image of cancer progression. Here's a normal epithelium. This is cervical cancer. And here's a sort of a low-grade neoplasia. It hasn't started to spread, but then it becomes a high-grade neoplasia, and then eventually it becomes invasive carcinoma, and it can escape, and the cells start to migrate, and eventually if they make it to the bloodstream, they can travel to a different part of the body. And so here's basically a nice little schematic of tumor progression. It all has to do with sort of clonal evolution. So you can get an accidental mutation in a gene, and all of a sudden that will start, by and large, that's not going to cause a big problem. However, if it gets an additional mutation, it can start to proliferate, and then eventually the proliferation is going to become dangerous and it can start to spread. Now there's a number of different ways in which things can happen to a cell to cause it to become cancerous. So, you can have genetic and epigenetic changes. So a genetic change is pretty simple to understand. You'll have a gene, you'll get an accidental mutation in it, and then every time that cell divides, it's going to copy that mutation. So then you'll have many cells with the same mutation. You can also get epigenetic inactivation, where there's some sort of aberrant signal that leads to heterochromatin packaging, and then all of the heterochromatin reader-writers will ensure that all of the subsequent cells in cell division are packaged into heterochromatin. And also you can have DNA methylation. And these two are pretty much related, for all intents and purposes. So in other words, you can sort of lose like a tumor suppressor through a loss of function mutation, or a tumor suppressor could technically be packaged into heterochromatin and silenced, and then it's unable to do its job. All right, if you can't suppress the tumor, it's more likely then you'll gain a mutation in like an oncogene or something like that, that then will cause the cell to proliferate even further. So karyotyping is a great classic way to sort of look at what's happening to the chromosomes. And so here's a karyotype slide, and then they can stain them. You can sort of look here that there's all kinds of translocations that have occurred in these. They're not just stained in your nice sort of color. Wish the book had a slide here with the normal one, but you guys can go back to the karyotyping slides that I covered before and just do a quick comparison. And one of the things here is if you count the number of chromosomes, here there's 48 instead of the normal 46. Okay, so we covered apoptosis in the last lecture, and we covered the cell cycle previously too. And so what you end up having in your body is basically this constant flux of cell division and normal apoptosis, all right? And that leads to homeostasis. So if you have normal cell division and normal apoptosis, as these some cells divide, some cells will die, and you have homeostasis. If you have increased cell division and normal apoptosis, the increase in cell division will start to produce more and more cells, and you get the formation of a tumor. Okay, The opposite could happen where you can have normal cell division and decreased apoptosis, in which case the cells are dividing but they're not dying as they should, and that can lead to a tumor. When you have the combination of increased cell division and decreased apoptosis, that's when things really start to go awry. Okay, so one of the things that they've really sort of found in the past decade or so is the importance of cancer stem cells. And so if you took a rat, this is a FACS cell sorting of rat glioma tumor, and one of the things they found is these are all of the tumor cells up here, but here's also some tumor cells down here. These are considered tumor stem cells, okay? And the thing with cancer and why it's so hard to treat is these cancer stem cells, they can self-renew, and they don't grow as fast, so they're harder to kill, right? Most of the chemotherapies that are used at this point in time sort of interfere with the cell cycle and things like that to just basically arrest the cell and cause it, to stop proliferating. However, cancer stem cells, they don't really proliferate that much. They're much sort of slower proliferating cells, and so they're much harder to kill with standard chemotherapies. That's why you can sometimes beat back the tumor, but then and the patient is in remission, but then the cancer comes back again a couple of years later when they're not on the chemotherapy. And here's basically how it works. You have a normal stem cell that's undergoing self-renewal. This is actually a relatively slow process. And that normal stem cell will eventually turn into a transit amplifying cell, which then becomes sort of, the cells that it needs to replace. So you can have a mutation or an epigenetic change in a stem cell, and that can lead to a malignant stem cell, or the transit amplifying cell can have a mutation or an epigenetic change that sort of cause it to go back to being a malignant cancer stem cell. And then this will undergo self-renewal, and then it becomes transit amplifying cells, and then all of a sudden you have basically a small little tumor with cancer stem cells and cancer transit amplifying cells. And once again, the normal tumor cells are pretty easy to kill, and even the transit amplifying cells are pretty easy to kill, but the cancer stem cells are much more difficult, making it a much harder disease to treat. Okay, here's the steps in metastasis. So normally you have an epithelium with your basal lamina, and then a cell can acquire a mutation, and that'll grow into a benign tumor in the epithelium, and then all of a sudden some more mutations may occur, and then it becomes invasive and enters the capillaries. Then it can sort of spread through the blood system, and then it'll eventually attach to the the wall of the capillary, and then it can invade into a cell and then start to divide and colonize, and then you have full-blown metastasis. Here's an example of that. This is actually an image of a lung, but it has colon adenocarcinoma sort of infested in there. So once the cancer metastasizes, it travels to different regions of the body. Okay, and in this instance, you have colon cancer in the lung. Okay, I'm sure you guys have all had the Ames test at some point in time, but I'll go over it quick. This is just a simple assay to tell whether or not you have a mutagen. And if you have something that causes mutations in your DNA, it's much more likely that you're going to end up getting cancer. And so what you end up having is a test compound. This is your potential mutagen, and you mix it with a culture of Salmonella that has a point mutation in histidine-dependent biosynthesis pathway. Okay, and then you also mix that with a liver extract, a homogenized liver extract, which has a lot of this enzyme called cytochrome P450, which I'm sure all the tox students are familiar with. Okay, and so you mix this together, and then you plate it on a plate lacking histidine, and technically none of these salmonellas should be able to grow because they lack the ability to synthesize their own histidine. Okay, now occasionally you'll get a single revertant that'll pop up on the plate, and it can grow, and it'll be fine, you know, because spontaneous reversion does occur on some level. But if you incubate it with a mutagen, that inactive histidine gene is more likely to revert. Okay, so because the genetic changes will be that much greater, and then you can sort of count the colonies and compare how many more colonies showed up with the mutagen versus the non-mutagen, and then you can determine whether or not something has the potential to cause cancer, or whether or not it has a mutagen. And here's a perfect example. This is aflatoxin. This is comes from an organism called Aspergillus ningulans, and it's just a filamentous fungus that was often, you know, on legumes and other things like that. And so if you happen to eat mold, what ends up happening, this product, aflatoxin, gets reduced by cytochrome P450, and then it actually covalently attaches to guanine and DNA. Okay, and when this eventually gets removed, what ends up happening is you have a G to T, a G to uracil change, which then all of a sudden, when it gets copied, it tends to become, it results in a GC to AT mutation, right? Because once this thing gets sort of removed, you're sort of like left with what looks like uracil there, I believe, and that's how that whole thing works. And so you end up with a lot of GC to AT mutations. And this actually happens in the liver. So if you've ever had, or if you've ever eaten aflatoxin, and you have sort of it covalently attached to DNA, and you have sort of hepatitis C or hepatitis B, your chances of getting hepatocellular carcinoma is astronomically high. Your chances are very, very high. Okay, so viruses, as I just sort of said, with the hepatitis viruses, they actually can contribute to cancer, and there's many others, Papillomavirus, the herpes simplex virus, and a couple of other ones, some RNA viruses. And the way this works is it's oftentimes indirect, and we'll get to that in a second. I guess this is the next slide where we sort of look at mutations. Okay, so you can have two things that can happen on the DNA level. You can get, you know, a normal cell, and then a simple mutation which causes a sort of gain of function or an activating mutation. Can happen in an oncogene, and all of a sudden this thing is sort of primed to become a cancer cell. You can also have loss of function mutations where you sort of mutate a tumor suppressor, and once you have the tumor suppressor sort of mutated, the chances are it can occur on another one, and then all of a sudden this is on its way to becoming a cancer cell. You can sort of see cancer cells, they grow, you know, indefinitely, and a lot of times you can culture them in the lab. And so normally, normal cells will undergo this thing called contact inhibition, where once the cells touch, they sort of stop growing. But as long as there's plenty of media around, if you have, you know, a mutation in like RAS or something like that, it'll actually create a mound of cells. And some of them will start to float off and things like that. So, you know, but so in general, these cells, you know, don't respond to the contact inhibition like normal cells would. Okay, so here's how things can happen. This happens to be with the RB gene, the retinoblastoma gene, and it's our universal regulator of the cell cycle, and we looked at it when we were covering the cell cycle, and I told you I would talk more about it when I got to the cancer lecture. And so normally what happens, you can have a healthy individual. When the cell divides, every now and then you'll get a mutation in the RB gene. But it's fine because you have 2 copies of it, not a big deal, you won't get a tumor. However, you can inherit a gene, the RB gene with the mutation, and then if you inherit the gene from one of your parents with the mutation, what ultimately will happen is the next time if the other gene gets mutated, then you're going to have excessive cell proliferation, and that's going to lead to retinoblastoma. And you can also have non-hereditary retinoblastoma, where you basically get normal, healthy individual, where one cell happens to get a mutation in the RB gene, but then if that cell goes on and it acquires an additional mutation in the RB gene, you'll get the formation of retinoblastoma and increased proliferation. We'll look how that works in a second, but here's some of the different mechanisms where you can lose the good copy of the RB gene. Okay, so you can inherit one chromosome with a mutation in the RB gene and then a normal RB gene, and you can just lose one of the chromosomes through non-disjunction, and then you're left with an inactive RB gene, especially if that chromosome then duplicates itself, then you have 2 inactive ones. You can get a mitotic recombination between these 2 chromosomes up here, so that both chromosomes then have the mutated RB gene. You can get a gene conversion, or you can get a deletion where you lose the RB gene, or a point mutation where the RB gene, and then ultimately in all of these instances here, you don't have a functional RB gene, and so that can lead to retinoblastoma. So the second you lose the tumor suppressors, all of a sudden cancer can sort of pick up and grow. You can also have tumor suppressors silenced, right? And so, you can get, here's your tumor suppressor gene, it can get a mutation, and then all of a sudden after a couple rounds of replication, then you have cells with both copies of your tumor suppressor mutated, and that can lead to cancer. You can get sort of heritable gene silencing through heterochromatin, and then that can either be spread to the other chromosome, or you can get a mutation. So you can have silencing and a mutation, or you can have both a genetic change and silencing. So all these different pathways can sort of lead to the cancer progression. You also typically need not only mutations in your tumor suppressors, but then you need basically activation of oncogenes. So you'll have a proto-oncogene, which is just normal, everything's fine, but then if you get some sort of mutation in the coding sequence that makes it hyperactive, you can basically get a hyperactive molecule. For example, the dominant RAS mutation it becomes a hyperactive protein where you can't hydrolyze GTP to GDP, and it's just active. You can get a mutation in the regulatory region, which then just produces more normal protein, and this can happen with certain transcription factors. You can get gene amplification, where now you just have more copies, and more copies of the genes then produces more messenger RNA of the genes, which leads to more proteins. Or you can have a chromosomal rearrangement where you get a new enhancer driving the expression, or you can get a rearrangement where you basically get a gene fusion, and so you get more of the mRNA. So lots of different things can happen. You can get mutations in intergenic regions that destroy loops, and then all of a sudden enhancers are there. We sort of talked about that when we were covering CTCF way back when. Here's just an example of MYC amplification. So here's a couple slides of it. You get MYC on chromosome A, and then you get aberrant replication. Then you get these sort of double minute chromosomes, and they can actually then refuse, and you end up getting a sort of an amplification of the number of MYC genes. So they can exist in sort of two different states, all as these double minute chromosomes, or they can refuse together. And there's another couple copies they've sort of integrated into different regions. And then once you've got MYC, you're basically wanting to drive the cell through different processes that are going to help lead it to cancer. So one of the great tools, and I sort of mentioned this earlier, is sort of comparative genomics, right? When we were talking about, whether or not cancers should be looked at as a tissue-specific disease or not. But one of the things you can do is basically see if you have sort of amplification. In the case with MYC here, you can do things where you basically take your tumor cell and you lyse it, and you isolate genomic DNA, compare it to normal tissue. You can fluorescently label them and then sort of put them on a microarray, and you can see that there's many, many more copies of MYC because this spot is red. This is getting largely replaced by DNA sequencing and RNA sequencing because it's much easier to just tell whether or not the MYC has many, many more copies of MYC. And these types of studies are now being done by the Cancer Genome Atlas, as well as hundreds and probably thousands of different labs all over the world right now looking at similar questions. But here's basically the expression changes where you get the gene amplification. Here's the normal location of MYC, and just some simple changes cause it to be, highly upregulated. And so, so even though a lot of the genes are the same, some are up, some are down, and you can sort of start to see global expression changes in cancer. And once again, cancer has sort of an additive effect, especially when it comes to oncogenes. So usually one doesn't do it. You need, activation of a couple oncogenes, as well as, suppression of tumor suppressors. And so here's if, the percentage of tumor-free mice, if they just have MYC. Eventually some of them get them, and RAS is a little bit more aggressive, but when you combine RAS and MYC, it becomes very aggressive. And so this is just sort of to indicate how cancer is actually additive. So the more mutations you get in oncogenes, and the more, sort of the gain of function mutations in oncogenes, and the more loss of functions in tumor, the more aggressive and the more additive of an effect there is. Okay, this slide is basically just a disaster. You guys should look at it a little bit. It's kind of confusing, but these are all the signaling pathways that can, and not all, it's actually just some of the signaling that can occur in cancer and where things can go wrong. Here you can see RAS. You also have, the death receptors. Here's your apoptosis regulators, BCL2, which we just covered. Here's P21, which we covered. P53 regulates P21. You've got anti-growth factors, Wnt signaling, the cadherins and beta-catenin. We've covered a lot of these different things here. And here's MYC down here in the middle. Here's the RB with EF2. P16 is upstream of it. And so all this can lead to different changes in gene expression and proliferation. No need to memorize this, but I do want you guys to sort of appreciate how complicated cancer and cancer cell signaling really is. And you can spend a lifetime working on just one single pathway, and there's a lot of people out there who have spent their lifetimes just working on one specific pathway here. Okay, so now we're going to look a little bit at RB in the cell cycle. So P16, that's a CKI, a CDK inhibitor. And so that normally is blocking cyclin D, which then prevents RB, and RB is there to prevent EF2. And this E2F is a transcription factor that helps the entry into S phase. Okay. And so what normally happens is you have P16 bound to the CDK, so you can't get a functional cyclin CDK formation, and that keeps RB in its active form, and it's basically blocking any S phase or entry into S phase. Okay, if somehow you lose P16, the cyclin CDK complex becomes active, and you phosphorylate RB, which then makes it inactive, and then you get active EF2, which then can start basically expression of genes needed for S phase gene activation. Okay, so you can see if you have a mutation that basically causes RB to be inactive, EF2 will be forcing, or E2F will be forcing the cell to go into S phase regardless of whether or not it wants to. So there's multiple signals towards proliferation, and we've looked at these. There's mitogens and growth factors, and the mitogens are needed to drive the cell cycle, and then the growth factors are needed for nutrient uptake. And when you get those two combinations here of the mitogens and the growth factors, you get cell growth and proliferation. And cancer cells actually need a lot of energy. And one of the key players here is mTOR. Okay, mTOR got its name basically because it's the target of rapamycin, which is a great chemotherapy drug. And for a long time they were using it and they didn't know what it was targeting. But here's what ends up happening. You'll get growth factors, activation of PI3 kinase, and then you'll get AKT. AKT will sort of activate mTOR, and that increases glucose transport, right? And then the increased glycolysis is going to lead to all kinds of things that the cell needs to, sort of grow, membrane biosynthesis and all of that stuff, right? So if you can block this pathway in the glucose uptake, you can effectively starve the cancer cells. Right, so cancer metabolism is sort of really area of high interest these days. So if you guys find this stuff interesting, you can probably find a lab that's doing it. Okay, there's also P53. We've covered P53 already. This is the sort of the Pinnacle tumor suppressor. Right, it's integrating signals from a whole bunch of things. You can have hyperproliferative signals that basically cause P53 to be active. DNA damage will cause it to be active. The telomeres get too short, or if there's a lack of oxygen, you'll get activation of P53. And when P53 is active, you can get cell cycle arrest, senescence, or apoptosis. Okay, and some of the targets of P53 are CKIs, which will sort of prevent progression into the cell cycle. Here's just a sort of an outline of abnormal, the formation of abnormal chromosomes. So you can have just a normal chromosome, and you can get a double-strand break that doesn't get repaired, and then if this goes through the cell cycle before it gets repaired, then you have sort of a broken chromosome without an end. Okay, in the next cell, this is going to be recognized as a double-strand break, and if this chromosome gets duplicated, you're going to get a fusion and then maybe a break somewhere else, and eventually you'll end up with just aberrant chromosomes. That's why when we looked at that sort of karyotyping experiment back a few slides, there were all kinds of different translocation events. Okay, and so in the absence of functional P53, this kind of stuff is going to happen all the time. So when it comes to viruses, here's just an example with what can happen with papillomavirus. So papillomavirus, normally if you get infected with the papillomavirus, you might get a benign wart or growth on your skin, pretty easy to treat. Compound W is something you can use, you just put it on. But eventually this virus, the chromosome of that virus, can actually integrate into your own gene. And if it integrates somewhere near an oncogene and cause activation, then you can basically, once you lose some tumor suppressors, you're probably going to end up getting a malignant tumor. And viral activation can also inhibit tumor suppressors. Okay, so here's normal RB in a quiescent cell. It's basically blocking E2F, and so the cell's not going to go into S phase. But if you have a viral protein E7, which titrates the RB off, then all of a sudden you have activation. And also, you can have similar things happen with P53. It's normal. It's going to lead to P21 transcription. It's going to arrest the cell cycle. However, if you have this viral E6 protein, it's going to bind to P53, and then all of a sudden you won't get any expression of the CKI or P16 to prevent entry into the cell cycle. Okay, so you just have increased proliferation. One of the big things with cancer too is what's called the EMT, or the epithelial-to-mesenchymal transition. And normally if you have sort of a tumor developing, and it takes a lot of effort for that cell to get into the bloodstream. But once it gets into the bloodstream, it can circulate, survive, and divide. There's plenty of growth factors and nutrients in the blood. And then it can get sort of stuck in a small capillary or something, which is easy. And then it can enter into a new tissue and colonize that area and then start to grow. This is a little bit more difficult. Okay, so you need to have escape, travel through the circulation, and then you colonize a remote site. And changes in expression with E. cadherin can actually facilitate the EMT and metastasis. Okay, so here's just some genetic lesions in colon cancer. There's plenty. If you go back to that signaling chart, you can find all of these players on there. But you know, RAS is an oncogene, proto-oncogene. Beta-catenin is also a proto-oncogene, and this will sort of affect receptor tyrosine kinase signaling, or Wnt signaling, or TGF-beta signaling. And then you can also have ones that affect DNA mismatch repair and others. Okay, so these are just the percentage that they occur in. So in the case with KRAS, it's found in 40% of human colon cancers. Here's just an image of a normal colon, and here's all these little polyps are colon cancer. And here's just the karyotypes of colon cancer. So this is a normal karyotype. You can see here there's 4 chromosome 1s instead of the normal 2. There's 5 chromosome 2s. So you can see it can get pretty messed up. And here's just a sort of an idea of sort of a schematic of how cancer can develop. You can have the loss of the APC gene, which then can cause a hyperplastic epithelium, in early adenoma. Then if RAS gets activated, then you have an intermediate adenoma. Then you sort of lose your tumor suppressors, and then eventually lose P53, you get carcinoma, and then invasion and metastasis. So the more mutations you get, the more aggressive the cancer becomes. And here's just a sort of a flow chart. I'm not going to go through this whole thing, but there's different mechanisms by which this can all happen. Here's 3 different cells and how they took 3 different routes to basically all become tumor cells. And these are just some of the genes that we saw in the table back here, and when and how they occur. But it's sort of the P53 one that really sort of is going to dictate what's going to happen when. Okay, so there's a specific two genes, BRCA1 and BRCA2, which are really common in breast cancers, but they can occur in other cancers as well. And what BRCA1 and BRCA2 are involved in is DNA repair. Okay, so when we covered what happens if you get a nick in DNA during DNA replication, right, and how you can get homology-dependent repair. Okay, there's actually two pathways for it. There's repair pathway 1 and repair pathway 2. We only covered just sort of the global one. But BRCA1 and BRCA2 are in repair pathway 2. Okay, PARP is in repair pathway 1. Or PARP is in repair pathway 2, and no, yeah, I had it right the first time, sorry. But generally in a normal cell, if you get a break during replication, this will repair itself, and ultimately you'll get repair. Okay, now if you're missing one of either BRCA1 or BRCA2, and you get this DNA damage, repair pathway 1 will take over and you're fine and the cell will replicate. However, if you inhibit PARP, which is involved in pathway 1 here, and you treat it with it, then there's actually no way to repair the cell. If you have a BRCA1 or BRCA2 mutation and you're inhibiting PARP, there's no way to really sort of fix this mistake, and there's no DNA repair, and the cell dies with fragmented chromosomes. Okay, so PARP inhibition is a great treatment if you have the BRCA1 or BRCA2 mutations. Okay, back onto the Philadelphia chromosome and Bcr-Abl. So here is chromosome 22, here's chromosome 9. Chromosome 22 has the BCR gene, and chromosome 9 has the Abl gene. When you get translocation in the formation of the Philadelphia chromosome, you get this Bcr-Abl protein, which then when it's transcribed is an aberrant tyrosine kinase, and then you get translation and this thing just runs amok. Okay. However, there's this great treatment now called Gleevec. Here's the chemical structure of Gleevec, which is a tyrosine kinase inhibitor for Bcr-Abl. And so here's the structure of the Bcr-Abl with Gleevec in it, and this completely inhibits it. And so here's just sort of a model of Bcr-Abl function. So normally in an untreated cancer cell, if you get the Bcr-Abl fusion protein, it's a hyperactive kinase that's basically going on and phosphorylating a whole bunch of things that gives signals for proliferation and survival, and you end up getting CML or leukemia. However, if you add Gleevec, which blocks the kinase activity of it, it can't phosphorylate its target, you get no signal, you get no leukemia. So this used to be one of the most deadly forms of leukemia, CML, but it's now one of the most curable forms because Gleevec is just so effective. And when it comes to treating cancer, we just sort of went over PARP inhibition and Gleevec, but oftentimes too, multi-drug treatment is turning out to be very effective. Okay, so oftentimes if you treat with one drug, you can reduce the tumor, but then, a rare mutant cell that's resistant to that drug will appear, and then the cancer will come back, and then it becomes that much harder to treat. But you treat it with drug B, that knocks it down, and one of the cells escape with another rare mutation, and then all of a sudden, treatment with either drug is useless. However, you simultaneously treat with two drugs at a time, you're not giving the cell the opportunity to actually become resistant to drug A or drug B. and that tends to work really well. Another thing that they're finding, and when we go over the circadian clock, I'll talk a little bit about chronotherapy. So chronotherapy is turning out to be very effective because up till now, chemotherapy drugs have basically been given when it's most convenient for the doctors, okay, and without taking the patient's underlying physiology into account. And so, which tends to be in the morning. People would go in, get their chemotherapy treatments in the morning, but that's when our metabolism is most active. Okay, so there's a lot of activity in our body, and that could be, that is basically taking in a toxin and making it even more toxic as it's being broken down. And so you end up having to give much more of a toxic compound to an individual to be effective because your body's breaking it down. Simultaneously, the targets that these various chemotherapies are going on may actually be expressed at certain times of the day when they're not effective. So for example, if you're giving someone a chemotherapy to target a specific drug or to target a specific protein or a specific pathway, but that pathway is not used until late in the evening, that drug's going to have to circulate in your system and while it's being broken down to treat it. So what they're basically finding with chronotherapy is that you can oftentimes use a lot less of a drug and get more effective treatments. So you don't get the nasty side effects and you have better outcomes. So it's just a really sort of a cool area that's sort of popping up these days. And something for everyone to keep in mind if you're heading into the medical profession. Basically decide when's the best time that a given patient should be taking their drug. If you don't know, you should try to find out because that could be very important.